everyone, and welcome to today's panel presentation titled Implementing a Real-Time PCR-Based Method for Sterility, Release, Testing, and ATMPs. This panel presentation is a part of the 9th annual event in the Microbiology Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Sartorius. For more information about Sartorius, go to www.sartorius.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits icon located on the far right of your screen and follow the process to obtain your credits. Now let's get started. I now present today's speakers, Dr. Alexandra Mueller-Scholz, Lead PCR and Microbiology Application Development Team at Sartorius, and Yasmin, Heynen, Molecular Development, Labor LS. For a complete biography on our speakers, the presenter tab is located on the left of your screen. Dr. Alexandra Mueller-Scholz and Yasmin Heynen, you may now begin your presentation.
So hello everyone, and thank you for the kind introduction, and also thank you for attending today's webinar on the implementation of a real-time PCR-based method for sterile release testing. And I will start with a short overview on the real-time PCR-based assay that was used by Labor LS during the study. So this is actually the current situation. So we have ATMPs, so-called advanced therapy medicinal products, which in many cases really have only a short shelf life, like 1 or 2 days until they need to be administered to the patient. But unfortunately, on the other hand, QC release testing using compendial growth-based methods take, for example, 14 days for classical sterility testing, or even 28 days until a contamination with mycoplasma can be ruled out completely. So, and that is why there's a really huge gap and the need for rapid testing. And that is also why Sartorius developed a real-time PCR-based assay for rapid sterility testing. So the method of choice is real-time PCR and involves at the beginning the actual test matrix, which in many cases are eukaryotic cells, for example, in a high concentration, like CHO or HEX cells. And in this matrix, there potentially you can find a small amount of contaminations, which should be detected with this method. So first step would be to extract the complete DNA of all organisms. And then next step would be the specific amplification of only the organism of interest, so our contamination, which is then detected via real-time PCR to receive a final yes-no answer. The actual assay which was used in the study is the microsat ATMP sterile release kit. It is able to detect bacteria and fungi contamination in only about 3 hours, and with a sensitivity of about 2.5 to 99 CFU per ml for all clinical relevant microorganisms. And the kit was comprehensively validated according to relevant guidelines like EP 516, USP 1223, EP2627 and USP 1071. One benefit or technology which is involved in this kit are highly specific TACMan probes. And these TACMan probes, they have a huge advantage. With the help of TACMan probes, you can reduce false positive signals due to an increased specificity, because not only the primers have to fit, but also the probes. So it's kind of a double proof concept. Furthermore, TACMan probes also reduce the risk of false negative results because they allow to do multiplexing. In this specific case, we have a duplex assay. In one channel, the FAM channel, we detect the actual target, so the bacteria or fungi contamination. And in the other channel, in the ROX channel, we have another set of primers and probes which detect an internal control DNA, which allow to tell whether the reaction is inhibited or not. The kit was comprehensively validated according to the guidelines, which are listed on the left side. And during the kit validation, we've covered, for example, specificity, equivalency, sensitivity, and also robustness and ruggedness. During the specificity testing, we've tested a broad range of different microbes of interest, so a huge panel of fungi and bacteria of clinical relevance, and demonstrated that we are able to detect them with a high sensitivity. Furthermore, we've checked that there's no cross-reactivity with, for example, relevant eukaryotic cell lines, which are present in the sample matrix. Another point was equivalency testing. Here we've used relevant bacteria and fungi species for spiking in a concentration low, close to the detection limit. And we've done PCR-based testing and the compendial growth-based sterility test in parallel at an external contract lab, and have shown that the results are equivalent. Furthermore, during our sensitivity study, we've tested a panel of microorganisms, so more than 20 microorganisms were tested with comprehensive dilution series. And here we determined a limit of detection for all relevant species between 2.5 CFU per ml up to 99 CFU per ml. And finally, during the robustness and ruggedness testing, we've tested different devices which can be used for real-time PCR detection. We've also shown that there's a low risk of false positives in terms of sample matrices and media ingredients. So for example, we've tested many different media and media ingredients as a starting matrix and demonstrated that there are no positive results at the end if there's no spike in the matrix. Furthermore, we've shown that we can successfully remove free DNA coming from dead microorganisms. So here with this tool, we kind of slight live dead discrimination. And finally, we also checked the tolerance to high cell backgrounds. So during the actual validation and also during extending studies, we've tested up to 25 million host cells in the background. And in the presence of such a high cell concentration, we were still able to detect small or low contamination concentrations, which is not too bad. So this I think gave you a pretty good overview on the actual kit validation, what we have done, and also an overview on the actual method and kit which was used during the study at Labor LS. And I will now hand over to Yasmin from Labor LS.
Thank you very much, Dr. Alexandra, for that wonderful overview. And hello from my side too. And thank you all for joining. So in my part of today's presentation, I want to show you a practical approach for the implementation of this real-time PCR-based method in a GMP environment. So the focus here today is on the practical aspects to consider and possible obstacles to overcome when implementing a rapid microbiological method. My part will be divided into two parts. So first of all, we will concentrate on how the implementation of a molecular biological method in a clean room class A can be achieved. So this includes a general technical setup regarding the equipment and the workflow, the choice of a suitable disinfectant as part of the contamination control strategy, an assessment of the suitability and purity of consumables, and the applicability of the assay within the cleanroom class A. And the second part focuses on the detectionability of the assay. The assay was performed including a panel of microorganisms in various media, for example, cell culture media and pharmaceutical products. But let's look at the starting point. In the left picture, you can see a cleanroom class A where a sterility test is being performed. The sterility test requires an aseptic environment. This means that there are very strict requirements on microorganisms as well as on particles. On the other hand, the right picture shows molecular biological techniques like pipetting or centrifugation. A rapid sterility testing using the molecular biological method real-time PCR combines those two working techniques. And to use a molecular biological method for a GMP-compliant sterility release testing, this real-time PCR must be performed in a clean room. And for this, we have to have a suitable setup and workflow. And this is the task I want to address in my part. So as I already explained, the cleanroom class A and B have strict limits for microorganisms and particles. Also, the real-time PCR method requires 2 centrifugation steps throughout the assay. Unfortunately, there is little to no documented experience with using a transportable centrifuge in a clean room. To avoid contaminations in the clean room that would negatively affect the sterility test, I assessed the impact of a centrifugation in a clean room. And for this purpose, I did a risk analysis. I sterilized a centrifuge, set it up in a clean room, and did a centrifugation. So I choose the position of the centrifuge that is marked here with the red square. In this position, the centrifuge has the maximum possible distance to the working area of the sterility testing, the clean room class A. And also in this position, the centrifuge is next to the ventilation systems that guide the air out of the clean room. So after positioning, I did the centrifugation. And in the meanwhile, I performed a microbiological monitoring of the environment and centrifuge and a particle measurement. The microbiological monitoring contains 17 measurement points, including the centrifuge, the floor, the table, and the air, and was performed before, during, and after the centrifugation. An aerobic and anaerobic incubation was performed for the measurement points. So after incubation, the molecular biological monitoring showed no growth of any microorganisms. Here I included 2 pictures of the plates from the monitoring. The particle measurement was evaluated using 6 particle sensors in the cleanroom class A and B. The sensors are able to detect particles sized 0.5 micrometers and 5 micrometers. The 2 graphs in my presentation that you can see here are the sensors that are located closest to the centrifuge. The graphs show the measured number of particles in the time before, during, and after the centrifugation. The red lines at the top of the graphs show the alarm limit of the particle sensors, and the blue lines show the measured number of particles over the time. In the left picture, you can see slight variations of the particle concentration, but these are within routine variations when operating in a cleanroom environment. But more importantly, all the points of the particle measurement showed no extension of the alarm limit at any time. So in conclusion, the microbiological monitoring confirms that the disinfection measurements are effective and also ensure the sterility of the centrifuge. The particle measurement shows that a centrifugation within a cleanroom class B is possible without disturbing the aseptic environment. But let's take a step back. What is the general benefit of this risk analysis, and why should we do a centrifugation in the cleanroom? So to begin, I designed a flow diagram with the testing procedure with the centrifugation steps outside of the cleanroom. So with a workflow like this, the assay is performed within the cleanroom class A, except for the centrifugation steps. So the assay starts in the sterility testing department with the disinfection of the material and the primary packaging of the samples. Then the assay starts with the extraction and isolation of the DNA from possible microbial contaminants. But in between that process, the closed samples have to leave the cleanroom because we have to perform the first centrifugation. So after that, the samples have to be disinfected again in order to continue the assay. But then there is the second centrifugation step. So again, centrifugation outside of the cleanroom and another disinfection step. So as you can see, this is a very time-consuming procedure, and it also increases the risk of contamination for the samples because they have to leave the cleanroom. And this could lead to false positive results. And false positive results mean economic damage for the pharmaceutical manufacturer because he has to discard the whole batch. But more importantly, the patient cannot get the pharmaceutical product he needs in time and has to wait again for the new batch to be manufactured. Especially short shelf life products are often produced in small batches and have a patient individual composition. So a failed sterility testing due to a high risk of contamination throughout the assay is severe. A better solution occurs with the successful risk analysis. So now the whole assay, including the centrifugation steps, can be performed inside the cleanroom. So again, the process starts with the disinfection steps of the material and primary packaging of the samples. The whole assay can be performed in the cleanroom until the real-time PCR can be performed. So it can be easily seen the workflow is now efficient, time-saving, and the risk of contamination is reduced. The second part of the practical implementation was to find a suitable disinfectant as part of the contamination control strategy. For this assay, microorganisms as well as environmental DNA can be a source of contamination. And on the market, there are many sterile antimicrobial disinfectants as well as cleaning agents to remove DNA and RNA contaminations. But unfortunately, there is no disinfectant that advertises to combine both aspects. So a sterile antimicrobial disinfectant with the ability to denature DNA is necessary. And for this, I did another risk analysis. I analyzed several sterile antimicrobial disinfectants to assess whether there is one that can also denature environmental DNA. So I incubated those disinfectants with lambda DNA and then performed a gel electrophoresis. And the results of this can be seen in this graphic. So first of all, we have the NC, the negative control, so the untreated lambda DNA, with its characteristic band pattern. Secondly, there are the results of the disinfectants 1 to 4. The samples 2 to 4 still show the characteristic band pattern of the lambda DNA. Only sample 1, the disinfectant with the hypochlorous acid, could successfully denature the DNA template. So as an additional test, I wanted to assess whether the treatment with the disinfectant with hypochlorous acid leaves any DNA fragments that can be amplified with our real-time PCR assay. So for this, I incubated Escherichia coli DNA with various volumes of the disinfectant with hypochlorous acid, and then performed the real-time PCR. The results of this can be seen in this amplification curve. So first of all, we have two curves with exponential growth. These are the positive control of the assay as well as the untreated DNA. And then we have the remaining linear curves. These are the negative controls of the assay as well as the DNA treated with the disinfectant. So the linear curves represent that no DNA was amplified, and therefore the given DNA template was denatured. In conclusion, the disinfectant with hypochlorous acid can successfully denature DNA and also leaves no fragments that can be amplified with this assay. The third part of the technical setup addresses the suitability of consumables. When performing a real-time PCR, the most commonly used consumables are PCR stripes or a 96-well plate. And at the beginning, I could detect contaminations in the negative and media controls in PCR stripes, regardless of how high my safety measurements were. So for a general assessment of the suitability of consumables, I analyzed the same samples using PCR stripes and a 96-well plate. And the results can be seen in this graphic. So we have the green curve that show the positive control of the assay, the blue curves that show the negative and media controls in PCR stripes, and the red curves that show the exact same samples in a 96-well plate. And additionally, I listed the CT values of the assay. The contaminations in the PCR stripes showed a CT value of 35. The same samples in the 96-well plate showed no CT, so a negative result in the real-time PCR. And this gives an indication about the suitability and purity of the consumables, even though they are both labeled as DNA-free. The high sensitivity of the assay can detect contaminations in consumables that can lead to invalid test results. So please keep in mind, when implementing a new method, consider the suitability and purity of the consumables. Now, after implementing the technical setup, I performed the assay as it would be for a GMP-compliant assay in a clean room. So for this assay, I used PCR-grade water. The results of the assay can be seen in this amplification curve. So again, we have curves with exponential growth. These are the positive controls of the assay, bacteria as well as yeast and molds. Then we have the remaining linear curves. These are the negative controls of the assay as well as the PCR-grade water. So the remaining curves again show no DNA was amplified, and therefore there was no microbial contamination. Additionally, I performed a microbiological monitoring and a particle measurement that showed no microorganism growth nor an increasing particle concentration. So with the results of the technical setup, we can say that the applicability of the assay within a clean room could be demonstrated, and the prevailing clean room conditions can be maintained. The established testing procedure with the centrifugation steps inside the clean room allow an efficient workflow and reduces the risk of contamination. And lastly, it is very important to consider the suitability and purity of consumables to avoid false positive or invalid results. In the second part, I confirmed the detection ability of the assay by detecting a panel of microorganisms as they are described in the pharmacopoeia. So the panel contains of 4 bacteria, a fungi, and a yeast. The concentration of the microorganisms was chosen to be 99 CFU per extraction. This concentration is also defined in the USP chapter 1071 for a risk-based release of sterility tests for short shelf life products. So I performed 6 extractions of each microorganism and analyzed those in technical duplicates. Additionally, I listed here the CT values. So as you can see in the results, we had a complete detection of 12 out of 12 microorganisms for this assay. And also you can see the CT values for the further comparison with the next assays. Because we are currently applying the assay on different pharmaceutical products. The aim is to assess the applicability on the assay on different matrices, here pharmaceutical products. So the first example shows peripheral blood mononuclear cells in a concentration to 10 to the power of 6 cells per extraction in cell culture media. Due to small batches and small sample volumes of most short shelf life products, these experiments could be performed with 4 replicates for each microorganism. The results again show a complete detection of all 4 replicates for all 6 microorganisms. Again, the CT values show a similar range compared to the previous assay. So now we can say the assay is applicable on samples like here blood cells. In the next assay, we included chondrocytes in cell culture media. The chondrocytes had a concentration of 10 to the power of 6 cells per extraction. Chondrocytes are cells of the cartilage tissue. So the provided samples represent a precursor to the final pharmaceutical product. The sample volume again allowed a number of 4 replicates. The results again show a complete detection of all replicates for all microorganisms. And the CT value show again a similar range compared to the previous assays. And lastly, I performed the assaying using chondrocyte spheroids. These samples now represent the final pharmaceutical product because now the chondrocyte cells have developed into a 3-dimensional cartilage tissue. The sample volume now only allowed analysis in duplicates for each microorganism. But again, the detection was for every replicate in every microorganism completely. And also the CT values show a similar range. So in conclusion of all the assays I performed, the test is applicable on pharmaceutical products, but for a GMP compliant matrix suitability testing, the assay should be performed using more sample replicates for each microorganism to better assess the applicability and robustness. Additionally, the matrix suitability testing should not be only performed using the panel of 6 microorganisms, but also environmental isolates of the manufacturer's monitoring and microorganisms from failed sterility testings. So lastly, I want to present you an overall conclusion of my practical implementation. As I already explained, the assay is applicable within a cleanroom class A regarding the established technical setup. The method can serve as a risk-based rapid sterility test for short shelf life products, and the matrix suitability testing should use appropriate microorganism strains in order to ensure a successful application of this technique. So thank you very much. Here you can see our contact details if you want any further information about this assay. And if you have any questions, we are very happy to answer them.
Thank you, Dr. Alexandra Mueller-Scholz and Yasmin Heynen for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. All right, let's get started. Our first question here asks, How much time can be saved when performing the centrifugation steps with the clean room?
So each disinfectant steps takes up to 45 minutes. So in summary, with the 2 centrifugation steps we have in the assay, we have 2 disinfection steps. So that's in total 1.5 hours of time that can be saved when you perform the assay in the clean room.
Great, thank you. We've got some great questions coming in from our audience. So this next one, Yasmin, is for you as well. Can the assay also be performed in an isolator?
So in general, yes, the assay can be performed in an isolator. We also have been doing some experiments on this, but we have seen that most standard isolators are not designed for molecular biological assays. So for example, you have to have a separation of the workflow, which is not quite possible in an isolator because there is little space. And also you have mostly large gloves that are very thick. So when doing a molecular biological assay, you mostly have to pipette small volumes, and this is not quite possible with those gloves. So an option would be to have individualized isolators for the use of molecular biological assays.
Great, thank you. Now, Yasmin, why do you use 2 safety workbenches in the clean room for the molecular biological assay?
So that's a thing you do when you have molecular biological assays. So in these assays, there is a high risk of contaminations of the reagents and controls. So for example, the PCR is a very sensitive method, and there is a high risk of contaminations leading to a false positive result. So when doing a molecular biological method like this assay, you have to separate these the workflow in separate areas. And that's why in the clean room, we have used several workbenches to separate the workflow.
Great. Well, it looks like we have time for one more question, so we'll wrap up after this one. And this final question asks, do you have any recommendations for testing the suitability of the consumables?
So first of all, I would recommend to get in touch with the manufacturer if you're not sure if the consumables are DNA-free. Most of them, they can give you a good answer if they are. But if that's not enough for you, I always recommend doing experiments on your own. So like I showed in my presentation, I took several consumables and performed the real-time PCR. One important note on this, you have to use a matrix, like I did cell culture media, that is DNA-free and sterile. So with doing several experiments, you can find out during your implementation if the consumable you want to use is applicable.
Thank you, Yasmin. Do you have any final comments for our audience?
So thank you very much for these great questions, and I would be very happy to hear some more from you. I thank you that you're so interested in our presentation.
Great. Well, thank you again, Yasmin, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Sartorius, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until September 7th, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, goodbye.